The cell nucleus is a membrane-bound structure that contains the cell's hereditary information and controls the cell's growth and reproduction. It's the command center of the cell and is commonly the most prominent organelle. It's bound by a double membrane called the nuclear envelope, and in between those layers is the nuclear lamella, which provides structural support and maintains the shape of the nucleus. Inside the nucleus, you have genetic information in the form of chromatin, which condense to chromosomes during cell division, and that carries your genetic information. The nucleolus is a dense region within the nucleus and is composed of RNA and proteins. Its number one job is to make ribosomes, and it does that by attaching itself to the ribosomal DNA genes in your DNA, in your chromosomes. It's the largest structure in the nucleus, and we're finding in recent years that it does more than just make ribosomes. It also makes other particles for signal recognition and communication, and it is actually the coordinator of the cell's stress response, which is relatively new information. The ribosome is a complex molecular machine found within all living cells that serves as the site of biological protein synthesis. They link amino acids together in the order specified by messenger RNA. They consist of two major components, the small subunit which reads the RNA and the large subunit which joins the amino acids together to form a polypeptide. Each subunit is composed of one or more rRNA or ribosomal RNA molecules and a variety of proteins. But beyond that, prokaryotic and eukaryotic ribosomes, while they both have them, they are not the same. Eukaryotic ribosomes are larger, and there's some good benefits to that for us. Say, for example, the antibiotic tetracycline, it works by stopping the ribosomes working in bacteria, but they don't actually hurt our ribosomes at all because they can't recognize them. So that's very helpful for us. Another difference is the difference between bound and free ribosomes. Free ribosomes are floating in the cytosol, and they make material that will stay in the cell. Bound ribosomes attach themselves to membranes, be it the endoplasmic reticulum or the nuclear membrane, and they make materials that are destined to leave the cell or at least become external to the cell. So free ribosomes make things that stay, bound ribosomes make things that leave. And I know that's a little bit backwards, but it's true. The endomembrane system is a series of organelles that work together in the production, packaging, finishing of proteins and molecules that are destined to leave the cell. And it's made up of several organelles that we're going to look at over the next couple of slides. The smooth ER is one of two types of endoplasmic reticulum, and it is a long network of a folded tube-like structure. Its main job is biosynthesis. It produces lipids and phospholipids and steroids and sex hormones. So cells which secrete these products are going to have a lot of smooth ER, testes, ovaries, sebaceous glands. Other jobs include carbohydrate metabolism, basically the breakdown of glycogen into glucose in the liver. It's going to detoxify drugs and poisons like alcohol and barbiturates, and it's going to be essential for muscle contraction as it regulates calcium. The rough ER is a convoluted but flattish sac, so it's more flat than the smooth ER, and another major difference is that it's studded with ribosomes. Ribosomes, why are they there? Well, ribosomes produce proteins, so the rough ER is going to work with ribosomes to make proteins, to modify them for the ways that are necessary. For example, we need glycoproteins, so I can add sugars to the glycoproteins in the ER to be used in your cell membrane. A ribosome only binds to the rough ER once there's a specific complex, so the bound ribosomes can leave the rough ER and come back. Sometimes they stay connected, but they don't have to. So the membranes made in the smooth ER and the proteins made in the rough ER can be sent to the Golgi apparatus. They're going to go as small bubbles of membrane called vesicles, which we'll talk about more in a bit. They will fuse on the cis side of the Golgi apparatus and be modified and changed and have signal molecules added as it passes through the layers of the Golgi. When it reaches the other end, the trans end, it'll be finished and it'll be marked almost like a barcode at UPS, and it'll bubble off into another vesicle. So it's like the trucks at UPS. So cells that specialize for secretion, endocrine glands like the pituitary, the pancreas, um, exocrine glands like digestive enzyme producers in the stomach, mammary glands, sweat glands, they're all going to have a lot of Golgi bodies. Lysosomes are small vesicles full of hydrolytic enzymes, which are enzymes capable of hydrolysis or digestion. So that's their main job, breaking down biomolecules like carbohydrates or lipids, or the example on the right is an entire mitochondria. But in the last couple of years, we found out that they're involved in a lot of other functions. Homeostasis of the cell. If your membrane is burst, the lysosome is what will fix it. Apoptosis or programmed cell death use lysosomes as part of its pathway. Cell signaling plays a role there as well. But if they don't function correctly, we end up with a problem. So Tay-Sachs disease is an example of this, where lysosomes in the brain of children cannot digest lipids. So the lysosomes fill up with lipids and eventually result in the child's death. The peroxisome is a specialized structure that's job is to break down fatty acid chains and amino acids, because these things are too big for a mitochondria to break down the way they are. It makes it into smaller pieces so that they can work with it. It has a secondary job of detoxifying things like alcohols and poisons by using hydrogen peroxides to oxidize them. About 25% of the alcohol humans drink is detoxified in this way. But that also means that they produce hydrogen peroxide, which is dangerous for you. But they do have enzymes called catalase that is able to break it down and make it safe. So vesicles are small organelles consisting of fluid enclosed by a membrane, and vacuoles are just larger ones containing mostly water. They're made from membranes, from the ER, the Golgi, or the plasma membrane, depending on where they're coming from. And it's all going to be about organizing and separating substances. Some of the major types are the large central vacuole of the plant, the transport vesicles that move material around in the endomembrane system, secretory vesicles in your neurons allow you to release things at the synapses. The picture on the bottom right is a contractile vacuole in protists, allowing it to maintain its water balance. You have transport vesicles, like we said, which move materials around, food vacuoles, which are for storage, which will eventually fuse with lysosomes for digestion. So overall, vesicles are organizational, separating things out and making them useful. The endomembrane system is a series of organelles. The nuclear envelope is connected to the rough ER, so material made there can flow out. Membranes produced and proteins produced by the ER will go to the Golgi, which will modify them and make them ready for transport. Lysosomes can take those vesicles and digest them if necessary. Transport vesicles will carry them to the plasma membrane for secretion, and the plasma membrane will fuse with that vesicle, releasing the materials, secreting them into the outside. So while it's a long series of organelles, it has one overall function, which is an important process to remember.